Good morning, everyone. It is really my great pleasure to be here today. And I've been to the airport many times, but this is the first time I can visit this city, and I was so impressed. Beautiful, and people are very friendly too. And also, I want to thank organizer, particularly Michael, and also Inez for inviting me here and help with all the logistics. So if we talk about the goal 2 of sustainable development goal, there are 3 aspects of it that you need to achieve food security. At the same time, you improve the nutrition and also promote sustainable agriculture. And I want to focus in the first part of my talk on the hidden hunger. Hidden hunger is the micronutrient deficiency, and globally, about 1.6 billion global population is actually anemic. And around half of it is because iron deficiency anemia. And at the same time, the zinc deficiency, which is causing the stunted, immunocompromised, affecting around 165 million children globally, which is 1 out of 5 children. And if you look at the brain of this stunted infant, which is around 2-3 months old, each gold represents a fiber tract, and the long thin fiber transmit information to different neurons, muscle, and glands. It is apparent how much denser if you children under 5 years old get enough zinc. So what the young ladies is make sure that your children have sufficient intake of zinc. Stunting is not only because of the zinc deficiency, but about 50% of it about zinc deficiency. And then there are 2 different concepts which is, should be complementing each other. One is, of course, we go for the diverse diet, but not everyone have the access for, you need a higher income or lower food prices. But when you apply lower food prices, the farmers were so unhappy. Why do it should be on their sake that you get a better price for the consumer, but that is the main aim. But if you can make the food staples increase the density of nutrients, so at least you put the baseline of zinc, then which is we call the biofortification through genetic approach, or if you improve the fertilizer, then you give the baseline of the needs. And our aim was to develop healthier rice with high iron and zinc rice to be combined with the golden rice, the provitamin A rice, with the product concept to increase the iron concentration in the milled rice from 2 ppm to around 10 ppm, so around 4 to 5 times, depends on your background varieties. And zinc concentration more than 30 ppm in the milled rice, which cover about 50% estimated average requirement. And the yield should be within the range of the conventional counterpart. Our target country, the first 3 target countries that approved by the donor is the Bangladesh, Philippines, with potential immediate spillover with supportive regulatory empowerment in Nigeria and Indonesia. If you think about rice, then there are quite a lot of zinc and iron on the leaf and the rice plants, but it's not transported to the grain. So our approach is, we look at the number of approaches. So our approach was eventually, we use a nicotinamine synthase gene. So nicotinamine is involved in the long distance transport of iron and zinc. And it's also a precursor of the deoxymogenic acid, the phytosidophore, which is released in the rice roots, basically to make iron, which is often exists in an insoluble form, to be a soluble form. So that was to mobilize iron and zinc, either from the root or from the plant itself to the grain. And then we combine it with the iron storage protein, the ferritin genes, and we got it from apple. 1 apple a day keeps your doctor away. So this, we use two different promoter. One is targeted directly under the aileron, and the other one is a glutein A promoter. The other one is globulin promoter, which goes a bit deeper, because at the end of the day, you still get a lot on the brown rice, but the brown rice at the same time has a phytic acid, so iron and zinc is not necessarily readily available. So when most people still consume the milled rice in these countries. So to cut the story short, we developed thousands of events, we selected it, then so now we are already in the advanced stage of development that we have a multi-location testing in Bangladesh for the 2 years, and also multi-location testing in Philippines. And we select lines which has no yield penalty based on the statistically. And also we're very happy that eventually we got lines with 10 ppm iron versus the 2 ppm, the non-GM counterpart in Philippines. And then it's higher in Bangladesh because they have a long duration on rice compared to the Philippines. And so we are now on the last step. We are developing collection of regulatory science data for the dossier. This one is equally complex with developing the transgenic material itself, because you have this, for the golden rice, we have approval in US, Canada, Australia, New Zealand, and in the Philippines. So we had the experience from there. We learned from the private sector how they did it. And so you have to do basically a comparative safety assessment of this molecular characterization, compositional analysis, agronomic performance. Are there any difference than any intended trait? And then you have to do also comparative safety assessment on molecular characterization. If it's no, you always have, I mean, a lot of requirement. If we publish something, you don't need to have a very pretty southern and each element, but here you need to have a southern from each element, one copy, absence of plant backbone, stably inherited. So a lot of cost is actually on developing this regular, but you have to do it. So we are 90% on finishing this. So the current, our breeders stack it with golden rice, but this is with the breeding. Because if we do molecular stacking, then we have to get all the data as a new event. Okay, so that is part of the work which was a transgenic work, but we also use the technology like many other speakers earlier here to do gene function validation. For drought, we work with Michael Purganan and Amelia Henry at Erie to test it for drought. We using like constitutive drought inducible promoter, different gene family. And also the PhotoBoost project consortium, which is more on improving photosynthesis. Why do we have to do this? Because a lot of critics at the beginning about the GM, not why we have to do it, of course, because a lot of aspects on the climate change as well. But in the beginning of the transgenic development, it said you have to develop consumer traits, right? Because consumer doesn't know what's the, but then at the end of the day, at the same time, that farmers, if you want adoption, you have to attract farmers. If you say to farmers, okay, this is healthier, but you cannot sell it more expensive because this is for the poor, what is their incentive to grow your, unless it's a government program, right? So you have to combine with farmer traits, which could be the climate adaptation mitigation traits. So, and in the past, we have also done work on the like low phosphorus anaerobic germination tolerance was published and yield under low end. But in this case, we only use it for the identifying the gene, and then the final product is done by marker-assisted breeding. That's to avoid all the complexity of the regulation. But for iron and zinc, you cannot do that. There is not enough variation for particularly iron. Okay, so I used to be, before I worked on this, I was much younger. And then it takes you 12 to 14 years to work on the GM work. But on the other hand, if it's got adopted, you talk about 0.8 billion of population. So it's a tough work to do, but rewarding. But now we more embarked on the studies using genome editing to improve zinc in milled rice, and also we stuck it with the farmers' trade. So the title of mine was more actually on the genome editing work. So at IRI, we have a well-established transformation protocol and highly efficient gene editing pipeline for Asian and African indica rice. So indica rice, for those who are not familiar with rice, it is a long grain type of rice, grows more in the tropical area, while the ball type is more, and the only problem with indica is generally more recalcitrant than the japonica, which used by mainly publication use japonica rice. But eventually we developed a transformation capacity that genotype independent, and we transform popular rice in Asia, in Africa, Komboka, Samba Masuri Faro, Swarna Cerang, and MTU 1010. Of course, the efficiency varied from 5 to 100%. But we also developed a simple factor with knockout efficiency from 50 to 80%. And if we talk about why genome editing, that it is highly precise, 20 nucleotides, I think most people know here, guide sequence. It's affordable for minor crops and traits of minor commercial value, presumably that you work out, of course, the freedom to operate. And it democratizes science, equitable benefits, because you can work with resource-rich farmers, small, large hectarage. And faster development of new traits, reduce breeding time by nearly 2/3, eliminate linkage drag, so we can use more potential of the gene bank material to include them longer. But it doesn't mean that genome editing is one tool of the breeding. So you still need to breeding to get the final lines, advanced lines to be edited. And then when you got something, you also need to integrate it back to the breeding pipeline. And it has tremendous potential. And if you look at the current regulation, it used to be red, most of the area here. And even 2 weeks ago, I think it was the European Union in the Parliament, it started to be a discussion and more open that SDN1, SDN2 might be considered as non-GM. So we have now, like India have a clear regulation, Philippines is very clear, no foreign genes, non-GM, while in some countries SDN1, SDN2. So we decided to do the promoter editing on the OSNAS2, the gene that we have the overexpression before in the transgenic approach to increase the expression of this one. It's basically we aim several motifs there, and then we found a good result from one is a cis regulatory element known as Ariat was deleted, and then we check on these transgene-free lines, transgene-free lines over 3 generations, it's stable. And also, what is interesting, I mean, we got more increase on the zinc per plant, but at the same time, you got increase on yield. So in the grain itself, we got some increase, but it is not as, because you got dilution factor, right? If you got more yield, at the same time, you got more zinc. But interestingly, we got more panicle branching here, which, am I doing the same? And it has the panicle branching, more panicle branching, which result in on more grain. But of course, this is per plant. I mean, a breeder would always test it in the field before we claim higher yields. So we are going to do that. And I think the hypothetical network, the positive effect on seed number and panicle branching is because of the cytokinin level probably increase potential. But we have to prove it more in this case. And then, we got something that we didn't expect, that we got higher yield and some increase also in the zinc. But it's a lot, if you count the grain zinc per plant, it's more, but grain zinc in the actual grain is less. So we have to combine it also with other gene. And we also, this work has been published with target agronomic trait important to the farmer, which is on the bacterial leaf blight. And we also had to work on the Tungro disease. For the bacterial leaf blight, basically, this bacteria has a transcription activator like effector, which bind to binding element on the sweet genes. So we disrupted the binding element of multiple sweet genes. So this has been done through marker as well, that you have resistant gene, but using the genome editing, you can edit multiple sweet gene at the same time to get a broad resistance. So we did disruption of this element, effector binding element, and which cause inability of the bacteria to bind. So to actually told the plant to produce more sugar. And in the result shows that we actually got plants which is resistant. We haven't tested in the hotspot area, but in the screen house it was very promising. And another one is that we also work on the blast. Blast is also main fungal disease. So main fungal disease that in collaboration with CSIRO, we target blast disease in rice. The work was successful on wheat using the LR67 hexose promoter symporter gene, and also in barley, orthodox. So basically there is a resistant allele there, and the difference is only one base pair between the resistant allele and susceptible allele. So we were using the ideas if we can edit this one base pair from G to C to alter glycines to arginine, and we are hoping to get the rice blast similar to wheat using prime editing technology. The prime editing technology at the beginning, it's very hot topic, but the problem, it has low efficiency. So we use the incremental improvement. Actually, this was, we got, it's from publication. This was like efficiency increase when adding nicks in the transite. Also optimize the annealing temperature, addition of a viral nucleocapsid in the prime editor. And using two different constructs with two single guide and three single guide, we were very lucky to have 50 to 70% of T0 plants have the intended guanine to cytosine transversion, which I first thought maybe you did analysis wrong. How come that this could be 50 to 70%? But it was when it was verified using, initially we did the restriction analysis, then with Sanger sequencing, and also we got some homologous plant. And we did got, because we first test the approach using the transgenic approach. The transgenic approach worked, but in the transgenic, of course, you have the endogenous allele and the new allele. So that we got improved resistant to fungal. And this case would be, we only have the new allele, but we did get the phenotype, which is a bit in the tip. There is a leaf tip, a little necrosis on that. So we are actually going to test it, the next generation on the transgene-free one, to expose it on the different fungal. So I just want to close this with not really a conclusion for everything, but there's a value proposition for genome editing because it's relatively accessible, affordable, and available to public and small and private sector. We are also expanding our work on nitrogen use efficiency now, and also yield, but yield is more challenging. So eventually hope that this can be released in our target countries, which is encouraging. Like the first generation of the BLB rice now has been shared with the Indian partner, and the donor now would fund the Indian partner to test it on the different location. I think with that, I want to end my talk, and thank you very much, and you're welcome to visit our lab in the Philippines. I want to acknowledge also the member of the team, and also we have collaboration from CSIRO, Evans Lagunda team, Frommer, and team from Hendrik HHU. Field Rice, we have always have a good relation with the national partners, with Field Rice, Abdul Kader and team, Michael and team. That is for the specific work that we, the title is here, but we have many other partners for a different type of trade. Thank you.